You know, think about a house for a second. Like if you want a house to last for decades, you build it with this incredibly rigid, unchanging foundation, right?
Yeah.
Solid load-bearing walls.
Right. You expect it to be perfectly stable.
Exactly. But what if, to keep that house actually functioning, you had to simultaneously tear down and rebuild its load-bearing walls every single day?
While you were still living inside it.
Yes, while living inside it. It makes absolutely zero sense.
I mean, structurally, it's a total paradox of engineering. You're trying to get extreme physical stability, but also constant dynamic demolition at the exact same time.
Which brings us to our mission for today's deep dive. We're looking at a groundbreaking 2025 paper from Science by Heller, Kurup, and Zhuang. And they essentially proved that our brain cells are doing exactly that.
Yeah, it's wild.
We're unpacking this thing called the membrane-associated periodic skeleton, or the MPS. We're going to get into exactly how neurons maintain the structural strength to survive your entire lifetime while, constantly dismantling that same architecture just to do basic stuff.
And to really appreciate what Heller and the team found here, we have to look at the dogma they completely shattered. Like, for years, the scientific consensus was that the neuronal cytoskeleton, specifically this MPS out in the axons, was this highly stable, completely static scaffold.
Which, that makes intuitive sense, right? Think about what an axon actually is.
Yeah.
These are the long, delicate cables transmitting electrical impulses. Like a motor neuron's axon stretches from your spinal cord all the way down to your toe. That's a microscopic tube over 1 meter long.
Yeah, it absolutely needs a rigid protective cage. Otherwise, just normal physical movement of your body would shear it in half.
Right.
So the assumption was always that the MPS was basically the cell's rigid steel frame. And all the old experiments seem to back that up.
It resisted actin depolymerizing drugs. It turned over its proteins super slowly.
But the catch there, and this is the big but, is that all of those old conclusions were based on fixed cell imaging.
Right. Dead cells.
Exactly. Chemical snapshots frozen in time. You aren't watching the actual living system. But this new paper changes the game because they used super-resolution live cell imaging. They watched the living, breathing system.
Okay, so before we get into the actual demolition of this thing, let's draw a picture for you listening. Let's visualize the architecture of this cage, because the anatomy of the MPS is actually surprisingly beautiful.
It really is. The best way to picture the MPS is to think of a traditional wooden barrel, like an old-school wine barrel.
Okay, I'm picturing it.
So the architecture relies on 3 main parts. First, you have the metal hoops that encircle the barrel. In the axon, these hoops are formed by short, capped actin filaments.
So they literally create perfect rings around the inner wall of the axon membrane.
Exactly. And they are spaced with this incredible mathematical precision, like exactly every 180 to 190 nanometers.
Wow. And then linking those actin rings together, basically serving as the wooden staves of the barrel, are these beta-2 spectrin tetramers.
Right, these long spectrin strings.
Right. They stretch lengthwise down the axon, tying one actin hoop to the next hoop, which creates this amazing, highly ordered periodic lattice.
And then finally, to lock the whole system under tension, you have capping proteins, specifically adenosine and tropomodulin.
The nails.
Yes, they act as the nails holding the actin filaments closed, making sure the hoops don't just unravel.
Okay, so actin rings, spectrin staves, and adenosine nails.
Yep. It forms this breathtakingly perfect geometric cylinder.
And for decades, textbook biology assumed that cylinder was permanent. But the researchers developed this Cree-inducible knock-in mouse to challenge that.
Right. They genetically fused a fluorescent tag amnion green to the natural spectrin in these mice, so the staves of the barrel are glowing green.
Okay.
Then they cultured those living hippocampal neurons and put them under a live cell structured illumination microscope, or SIM.
And SIM is the technology that bypasses the normal limits of light microscopes, right? It gives us super-resolution video of living cells.
Exactly. They took images every 15 seconds over a 4-minute window.
And the footage is just astonishing. The
Right.
barrel is not static at all. It is literally constantly degrading and reforming in real time.
Over 80% of all the axons they looked at were doing this.
Wait, 80%?
Yeah. And in just that tiny 4-minute window, 20 to 30% of the entire length of the axon skeleton was actively dismantled and then rebuilt.
See, I have to push back on this because the physics of that just don't add up to me. Like, if evolution favors energy efficiency, this is a nightmare.
Oh, totally.
The ATP cost of rebuilding a third of your neural skeleton every 4 minutes? That's astronomical. And honestly, if 30% of this load-bearing wall is dissolving, Why doesn't the axon just collapse inward and sever?
Well, the survival of the axon completely relies on the localized nature of the remodeling. It's patchwork.
Oh, okay.
The entire axon doesn't dissolve all at once. What they mapped out was that these small, discrete patches of the MPS transition from a static state into this highly transient, fragile state.
I see.
And this local disassembly happens mostly in the medial and distal parts of the axon, out toward the synapses.
Okay, so it's less like tearing up the entire highway and more like repairing potholes one by one.
Oh, exactly right. The intact barrel structure around the pothole keeps enough tension to prevent the axon from collapsing.
Okay, that makes sense. So before we look at what triggers this, let's do a quick bullet point recap for you listening, just to lock this in.
Sounds good.
Number 1, the MPS is an actin spectrin adducent lattice, like a barrel. Number 2, we used to think it was a rigid, permanent cage.
Wow.
Number 3, live cell SIM imaging proved that it dynamically remodels 20 to 30% of its length continuously.
Spot on.
So, quick question for you to chew on. Think about a physical structure in your own life, maybe your bones or a city's bridges. How would you design a system to replace the structural parts without stopping the traffic? Keep that in mind.
It's a great engineering prompt.
All right, moving into segment 2. We have this localized demolition. What actually pulls the trigger on this? What orders the teardown?
So tracking down that trigger required a serious shift in their methods, because super-resolution SIM imaging is amazing, but it is incredibly slow.
Right, you can't use it to run high-throughput screening on dozens of different drugs to find the mechanism.
Exactly. So they came up with an ingenious workaround. They switched to a faster but blurrier diffraction-limited imaging technique.
Okay, so they couldn't see the perfect 190 nanometer rings anymore.
Right, but they could measure the fluctuations in the fluorescence signal using a mathematical tool called fluorescence autocorrelation analysis.
Which sounds like a mouthful.
It does, but essentially by tracking how much the glowing spectrin signal vibrated or shifted over time in a specific microscopic zone, they created a mathematical proxy for remodeling.
So high signal fluctuation just meant the barrel was actively falling apart and rebuilding.
Exactly. And that allowed them to rapidly screen a huge library of variables. And the prime suspect that popped out, intracellular calcium.
Calcium, of course. It's always calcium.
Always calcium. But they built a really elegant hierarchy of proof here. First step, chelation. They introduced a drug called B-apite, which acts like a chemical sponge.
It just aggressively brains up all the free calcium inside the cell.
Right. And the moment they sponged up that calcium, the structural remodeling almost entirely halted.
The MPS just froze.
Froze solid into its static state.
Wow. And then they ran the reverse experiment, which is honestly one of my favorite techniques in biology right now, calcium uncaging.
Oh, it's so cool.
They flood the cell with this specialized calcium compound that is totally inert, just floating there, until you hit it with a specific wavelength of UV light.
Mm-hmm.
And when they flash the neuron with UV, the compound breaks open, suddenly flooding the local area with free calcium.
And the response was instant. The local MPS barrel underwent massive, rapid degradation. So that definitively proved calcium isn't just correlated, it is the absolute non-negotiable spark.
It's the trigger. But, knowing the spark doesn't tell us where the match is being struck. Like in a living brain, where is that calcium coming from?
Right, because the neuron has its own internal calcium vaults, the endoplasmic reticulum, but it's also sitting in a bath of extracellular calcium.
And this is where it gets really interesting, because they tied the structural biology directly to the electrical firing of the brain.
Yeah. They started by applying TTX, tetrodotoxin.
The pufferfish poison.
Exactly. To totally silence the neurons and stop all action potentials. And when the electrical firing stopped, the structural remodeling dropped significantly.
It doesn't stop completely, right? Which implies there's like a baseline maintenance level.
Right, but the active electrical firing clearly drives the bulk of the teardown.
So they knew it was voltage dependent. Then they used specific pharmacological blockers to find the literal door the calcium was using.
Yep. They blocked the internal vaults, the ER, with a drug called thapsigargin.
And nothing happened. The remodeling kept going.
Kept right on going. But when they blocked specific voltage-gated calcium channels on the cell's outer membrane.
Specifically the N-type and L-type channels.
Right. They blocked those with drugs like omega-conotoxin and nimodipine, and the remodeling completely plummeted.
Which paints this crazy picture. The calcium driving this internal demolition is rushing in from the outside environment.
Exactly. Every time a neuron fires an action potential, or even just from spontaneous background electrical noise, these voltage-gated channels swing open. Calcium floods in, and the skeleton instantly destabilizes.
Okay, let's pause for recap number 2. Quick bullets. Number 1, they used autocorrelation analysis to rapidly screen drugs. Number 2, calcium is the absolute required trigger for the remodeling. Number 3, that calcium comes from outside the cell via N-type and L-type voltage-gated channels driven by electrical firing.
Perfect.
And a question for you. If neuronal activity, just raw electrical firing, increases this teardown, what do you think is happening to the scaffolding when you are intensely learning a new skill and your neurons are just firing rapidly?
It's basically liquid.
Exactly.
Yeah.
Okay, segment 3. We know the trigger is calcium. But calcium is just an ion. It's a signaling molecule. It can't act as a pair of scissors. It can't physically break protein bonds.
Right. It doesn't have enzymatic activity. It has to hire a molecular demolition crew.
And the paper lays out this brilliant dual pathway mechanism. It's not just one switch. To remember this, we can use the carry-R mnemonic. CAR calcium leads to adenosine PKC, which leads to rupture calpain.
I like that. CAR. So let's look at the first pathway in that mnemonic, the adenosine and protein kinase C, or PKC.
So when the calcium rushes in, it activates PKC. And PKC is essentially a molecular tagger.
Right. It immediately targets the adenosine proteins. Remember, those are the nails holding the actin hoops closed.
PKC adds a phosphate group to the adenosine. And that phosphorylation structurally changes the adenosine so much that it just falls off the actin.
It pulls the nails out.
Exactly. And without those caps, the short actin filaments become highly unstable. And they prove this by inhibiting PKC with a chemical called GFX, which stopped the remodeling.
And then they supercharged it with PMA, which caused the actin rings to just rapidly fall apart.
But pulling the nails is only half the demolition job, right?
Mm.
You still have the long spectrin staves holding the loose hoops together.
Which brings us to the R in your SAR mnemonic, rupture and calpain. At the exact same time calcium activates PKC, it also activates calpain, which is a calcium-dependent protease.
A molecular pair of scissors.
Exactly. And calpain's specific target is the spectrin string. So while PKC is pulling the nails off the actin, calpain swims up and physically cuts the spectrin tetramers in half.
And the evidence for this was so cool. They didn't just inhibit calpain with a drug MDL to watch it stop.
Yeah.
They actually engineered special antibodies that only bind to spectrin that has been physically cut by calpain.
Yeah, that was brilliant.
Under the microscope, they could literally see the fluorescent signal of those cut spectrum fragments perfectly correlating with the areas where the barrel was degraded.
The synergy here is just fascinating because by stripping the adenosine caps and loosening the actin rings first, the cell actually exposes the spectrin strings, making them way more vulnerable to being sliced by calpain.
Like loosening a knot before you cut the rope.
Oh,
And the researchers also noted that formins, which are proteins that regulate actin, also play a role in this whole dynamic state.
right. But it raises a really interesting evolutionary question. Why use two separate complex enzymatic pathways to take this down? Why not just evolve one master enzyme that destroys the whole thing?
I mean, I'd assume it's a fail-safe, right? If you just have one master switch, you risk a catastrophic runaway reaction where the whole axon just melts.
Exactly. Redundancy. By requiring both the phosphorylation of the caps and the cleavage of the strings, the cell gets exquisite dial-like control over the tension of the axon.
Okay, recap number 3. Number 1, calcium hires 2 demolition experts. Number 2, PKC removes the adenosine nails, destabilizing the actin. Number 3, calpain acts as scissors to cut the spectrin strings. Number 4, working perfectly together, they disassemble the lattice.
Nailed it.
So question for you listening. Why do you think the cell goes through the trouble of using 2 tools instead of 1? What advantage does that redundancy give the neuron in a stressful environment?
Always good to think about the evolutionary why.
Which brings us to our 4th and final segment, the ultimate why. We understand the anatomy, the calcium trigger, the PKC and calpain demolition crew. But what is the actual biological payoff here? Why spend so much energy to locally dissolve this cage?
It all comes down to one of the most basic things a cell has to do to survive. Endocytosis.
Eating.
Basically, yeah. A neuron has to internalize materials from the outside environment. It needs to absorb nutrients, take up signaling molecules, recycle its membrane.
But to perform endocytosis, the cell membrane has to physically bend inward. It has to invaginate, pinch off, and form a little internal bubble of vesicle.
Right. And here's the problem. The MPS creates a massive physical barrier to that.
Because if the inner wall of the membrane is glued to this rigid lattice of actin and spectrin, the membrane literally doesn't have the physical slack it needs to bend inward.
It's stuck. So the MPS isn't just the skeleton, it's basically an obstruction to cellular feeding.
Wow. So the rapid remodeling is the solution. The calcium spark triggers the demolition crew to snip a microscopic hole in the barrel.
Exactly. The local destruction creates a temporary window of flexibility. The membrane caves in, swallows the cargo, forms the vesicle, and moves it inside.
And the second that vesicle is clear, the neuron triggers a massive repolymerization event. It furiously rebuilds the actin hoops and spectrin stays to restore the cage before the axon gets mechanically damaged.
And they proved this functional link by tracking the uptake of membrane dyes. They watched endocytosis spike precisely in the exact microscopic zones where the skeleton was temporarily degraded.
That is just incredible. But the big picture here, the implications of endocytosis in a neuron, it goes way beyond just, absorbing food.
Oh, absolutely. Endocytosis is how neurons regulate synaptic plasticity.
Because neurons communicate by releasing neurotransmitters that bind to receptors on the next cell.
Right. And if a neuron wants to turn down its sensitivity to a signal, it has to physically swallow up and remove those receptors from its surface through endocytosis.
So this structural remodeling of the cage is acting as a master dial for communication. By tearing down and rebuilding this scaffold, neurons are constantly shifting their receptors around, tweaking how loudly they broadcast or how closely they listen.
And if you extrapolate that, it forces us to look at neurodegenerative diseases in a totally new light.
How so?
Well, we know genetic mutations in spectrin and other skeletal proteins cause severe neuropathologies. If this dynamic balance fails, like if the calpain scissors become hyperactive, or if the cell can't rebuild the cage fast enough, the neuron can't traffic its receptors or absorb nutrients.
The cell essentially starves, communication breaks down, and the axon dies.
Exactly. It is a very delicate, high-stakes molecular tightrope.
All right, final recap, number 4. Number 1, the MPS acts as a physical barrier to the membrane. Number 2, to absorb materials via endocytosis, the membrane must bend. Number 3, this localized calcium-driven demolition creates the slack needed to take in cargo, and then the cell instantly rebuilds the cage.
Perfect summary.
And your final question to think about. If the cell has to break its own skeleton to communicate, could a failure to properly rebuild that skeleton be the literal root of cognitive decline or neurodegenerative diseases?
It's a huge area for future research.
It just gives you this profound appreciation for the sheer mechanical genius inside your own head. We think of biology as like muscles flexing or lungs expanding, but every thought you have relies on billions of microscopic hoops and strings being snipped and tied back together in a fraction of a second.
It really does. And I want to leave you with one final, truly fascinating concept to mull over.
Let's hear it.
We established that this structural remodeling happens mostly out in the distal axons, right near the synapses where information is actually transferred between neurons.
Right.
And we know it controls receptor trafficking. So could this continuous microscopic cycle of physical destruction and reconstruction be the actual physical manifestation of how we update our memories?
Oh.
Right, like when you learn a new fact today or master a physical skill, are you quite literally tearing down and rebuilding the microscopic scaffolding of your mind?
That is an incredible thought to sit with. The physical architecture of our memories might just be this endless cycle of perfectly choreographed demolition. Well, thank you for taking this deep dive with us today. You will definitely never look at a static biology diagram the same way again. Stay curious, and we'll catch you on the next deep dive.